The correction of sickle cell disease. This presentation is brought to you by Group A4, Shannon Ivanchek, Alexa Dekbar, Haley Greenfield, and Taylor Gerals. So first, I want to introduce this disease for those of you who may not be familiar with it. Sickle cell disease, also called sickle cell anemia, is a disorder where red blood cells are disfigured and their function is reduced. Sickle cell disease is passed down from parents to children, making it an autosomal recessive trait. Hemoglobin, which is a protein inside red blood cells that carry oxygen, is responsible for causing sickle cell disease when an abnormal type of hemoglobin called hemoglobin S is involved. This happens because of the production of chains of beta hemoglobin that are not functional because beta-6 glutamic acid has been replaced by valine. Hemoglobin S changes the shape of red blood cells from the normal disc shape to a sickle or crescent shape. These crescent-shaped red blood cells carry less oxygen than normal red blood cells. They also can break off and get into the bloodstream and block pathways, which also prevent oxygen loss and prevents oxygen from getting where it needs to go inside of the body. Here you can see a normal red blood cell and then the sickle-shaped red blood cell, which is circled and looks like the crescent. Here you see a diagram of hemoglobin, which consists of 4 polypeptides, which all have an oxygen molecule in the center. Now let's talk about symptoms and effects and treatments of sickle cell disease. Sickle cell disease cannot be seen until an infant is 4 months old. Symptoms include fatigue, paleness, rapid heartbeat, jaundice, ab pain in children, and confusion. Eventually, the spleen will fail due to sickle cell disease. Life expectancy is now around 50 years, but used to be around 30 years. It has improved because of treatments, but these treatments are not definite. It is commonly treated by multiple blood transfusions and living a careful lifestyle. Taking preventative actions like drinking lots of water are also important. Most commonly, an organ failure or infection will result from sickle cell disease, and this will cause death, so many antibiotics are given to patients to prevent infection. Now that you are familiar with sickle cell disease, we are going to discuss an experiment that was done to try and correct the mutation which causes sickle cell disease. This is very interesting because sickle cell disease has been very elusive to scientists. It was the first genetic disorder in which a causative mutation was discovered, but this happened over 100 years ago, and no one has found a treatment to correct the mutation yet. So this experiment is called correction of sickle cell disease in adult mice by interference with fetal hemoglobin silencing. And basically what happens in this experiment was that mice were injected with hemoglobin A, which is adult version of hemoglobin, versus the fetal type of hemoglobin, which is considered to be healthy. And the goal of the experiment was to get fetal hemoglobin reactivated. And the way they wanted to do this was through BCL11A, which is a transcription factor that was used. In the discussion part of our presentation, we will go into detail about how this affects the production of fetal hemoglobin. However, on a side note, BCL11A is also the gene that encodes the protein that causes leukemia in some people.
The data found that a loss of BCL11A in adult mice reverses gamma silencing. BCL11A inhibitor allows for the switch of fetal hemoglobin to adult hemoglobin. This process is reversible, which is very important to note in this experiment. The switch from adult hemoglobin cells to fetal hemoglobin cells decreases the severity of symptoms of sickle disease. This is a logical explanation because the disease has not progressed when it is in the fetal stage. BCL11A null alleles were introduced into mice with sickle cell disease, as referred to as SCD. This is then called SCD-BCL11A. Part A shows magnified blood smears of the control, which are just red blood cells, sickle cell disease and SCD-BCL11A. As you can see, cells with SCD are misshaped, while cells with SCD-BCL11A are almost all normal. Part B shows that the lifespan of red blood cells is extended in SCD-BCL11A mice, as compared to mice with SCD. As shown, mice without SCD have a much larger lifespan than those with SCD. Mice with SCD-BCL11A live almost the same amount of time as mice without SCD. Part C shows a correction of the enlargement of the spleen in mice with SCD BCL11A. This shows that the weight of mice spleens that do not have SCD is roughly the same as mice with SCD BCL11A. Part D shows the expression of gamma globin and SCD globin quantitative reverse transcription polymerase chain reaction in the control, SCD cells and SCD BCL11A. It is shown that SCD BCL11A has the highest values. Quantitative reverse transcription polymerase chain reactions are used to qualitatively detect gene expression through creation of complementary DNA transcripts from RNA. Part E shows the distribution of fetal hemoglobin in red blood cells. SCD BCL11A has the most fetal cells as compared to red blood cells in SCD.
Many aspects of sickle cell disease and the research described in the article relate to our studies in this class. Sickle cell disease is caused by a point mutation in adult hemoglobin. The transcription factor BCL11A, when present, causes fetal hemoglobin to switch to adult hemoglobin. However, when BCL11A is inactivated, the hemoglobin remains fetal. When BCL11A is inhibited in mice with sickle cell disease, healthy fetal cells are produced and alleviate the effects of the disease. The data from this experiment indicates that BCL11A silencers can be utilized in future treatments of sickle cell anemia, which will now be discussed.
Interfering in silencing of the BCL11A is a difficult process to target as a therapeutic alternative for sickle cell disease. Repressing BCL11A in sickle cell anemia and beta thalassemia disorders can be accomplished by RNA inhibitory molecules, somatic gene transfer of short hairpin RNA molecules, as well as genetic screenings for the peptides that could inhibit BCL11A function directly or through disruptions in protein-protein interests. Loss of BCL11A enhances known fetal hemoglobin levels, so by inactivating BCL11A, fetal hemoglobin is silenced, which causes a decrease in the severity of symptoms for sickle cell disease, which can potentially be a therapy. In conclusion, we are now a lot closer to finding a definite treatment for sickle cell disease. And now to conclude our presentation, we have a clicker question. What is the cause of sickle cell disease? The correct answer is E.